Hello to all dear students. So in this video we are going to discuss about the last topic of your therapeutic drug monitoring subject, that is protocol for therapeutic drug monitoring. So the protocol goes this much. We have got two slides of side heading itself. So we are going to discuss about these all side headings one by one. Okay. So, starting with the first one, that is title of the study or project. Say you need to give an appropriate title for the project. Like, for example, if you're undergoing a study on, suppose, vancomycin in adult people, so you need to make a title like vancomycin dosing and monitoring in adults. If suppose you're studying the same drug in patients with some sort of renal impairment, then your title should be like vancomycin dosing and monitoring in renally impaired adults. Okay, so title should show everything about the thing what we are doing and in whom we are doing the thing. Okay. Next thing will be investigators. So basically we have got three investigators over here: chief investigator, joint chief investigator and co-investigator who will be a clinical research fellow. See, ultimately these all peoples are responsible for the design, conduct, analysis and reporting of a clinical study. They should have suitable experience and expertise in conduct of such studies in order to undertake same thing, the designing, the conduct, analysis and reporting of a study to required standards. So our investigator, they might be any medicine clinical pharmacist, or they could be nephrology clinical pharmacist, or they could be infectious disease clinical pharmacist, or they could be ICU clinical pharmacist and so on and so forth. Okay. Next thing is place of study, where you need to carry out your TDM work. So studies can be conducted in hospitals with good laboratories and qualified clinical pharmacists. Next thing is patient recruitment. Like, it is like on whom we should conduct TDM of a drug. Like suppose there is a lady who is suffering from ectopic pregnancy, and so as to remove the fetus from fallopian tubes, the doctors will be prescribing a drug known as methotrexate. Now methotrexate, it is a drug that have got a very narrow therapeutic index. Okay. So, in such case, such a lady with ectopic pregnancy will be recruited for the TDM study. Okay. Next thing is need for TDM. Like, why we need to carry out TDM? We need to carry out to establish a standardized pharmacokinetic approach for patients receiving drugs that are routinely monitored utilizing their serum drug concentration in any hospital. Okay. So basically TDM can guide the clinician to provide effective and safe drug therapy in the individual patients using the serum drug concentration. Like our example of methotrexate, since its therapeutic index is very narrow, if the serum drug level is subtherapeutic level, then it will not remove the ectopic pregnancy. And if the serum drug level is more than the therapeutically effective dose or the serum level, then it will produce life threatening toxic effect. Okay, so for this reason we need to carry out TDM work. Next thing is objective of TDM study, like why we need to study TDM, what is our objective. So basically we have got 6 objectives for studying TDM. Objective number 1 is obtaining maximum beneficial outcome of drug therapy by appropriate dosing of drugs. Objective number 2, providing medical advantage by reducing the chances of drug toxicity. Objective number 3, providing economic convenience to the patients by shortening their hospital stay. Objective number 4, monitoring factors like disease state, patient's characteristics, drug interactions, etc. in order to reduce patient variability. Objective number 5, dosage adjustment in patients with pre-existing hepatic or renal impairment and thus preventing drug accumulation in the body. And our last, that is objective number 6, providing an effective means of individualization of dosing of some drugs that have unpredictable dose response relationship. Okay, so these 6 are our objectives for studying TDM. Next is criteria for of the study. Okay, like why we need to do this TDM. What all are the criteria for studying TDM of any drug? Okay, so suppose a drug, the TDM procedure will be initiated once the pharmacy department got order of a drug that have got a very narrow therapeutic range, or therapeutic effect cannot be readily assessed by the clinical observation, or else another criteria could be if a direct relationship exists between drug or drug metabolite levels in plasma and pharmacological or toxic effects, or else another criteria can be if suppose large inter-individual variability in plasma drug concentration is being seen, or if a drug is following a non-linear pharmacokinetics, then we need to do such kind of TDM study. Or if a drug is following saturation metabolism, then also we need to carry out TDM. And lastly, if drugs used for life-threatening diseases, okay, means drugs are used for treating some kind of life-threatening conditions, then in such case we need to carry out the TDM study. See, now upon receiving the order for pharmacy to dose a specific medication, a pharmacist will assess the patient and collect relevant information necessary to appropriately dose or monitor the specific drug so as to achieve therapeutic drug levels and minimize any potential risk of toxicity. Such items of information it may include number 1, indications for therapy, that is type and site of infection for any antibiotic dosing or monitoring concern. Secondly, it should have age of the patient, next gender of the patient. It should also include height and weight of the patient. It should also include renal and hepatic functions, and it should also include the estimated pharmacokinetic parameters. Okay. Next is patient history. So patient history includes information about allergies, illness, surgeries, immunization and results of physical examinations and tests. It should also include all the currently and recently prescribed drugs, previous adverse drug reactions including hypersensitivity reactions, any over the counter medications including herbal or alternative medicines, and adherence to the therapy. Next thing is withdrawal of blood sample and storage. See, once all patient information, laboratory and clinical data completed, the pharmacist in charge start to follow sample guidelines and medication monitoring guidelines to monitor medication level, patient situation and follow up the patient during therapy duration. See, the laboratory and nursing staff will work together to determine the optimum time for specimen collection. For TDM to be meaningful, the patient should be in steady state on the present dose of the drug. In general, samples should be taken after drug dosing has continued for at least 4 half-lives. Unless otherwise specified, sampling times will utilize terms like trough means immediately before the next scheduled dose and peak that means 1 hour dosing. But in cases of suspected toxicity, waiting to attain steady state is not necessary and a random drug level may be collected to aid in clinical decision making. The laboratory department should deal with the drug level sample as urgent order, and results should be available in the system as soon as 1 to 2 hours max. So we have got various instruments like for measurements of drug level and for measurements of clinical parameters. Okay, we have got various instruments like HPLC and so on and so forth. Okay. Next thing is report preparation. The test results should be communicated as quickly as possible once it is verified by the senior laboratory personnel. The drug concentrations measured are generally they are reported in mass or molar units. But since most of the tests are done by biochemical methods, results may be in molar units and the laboratory should be able to readily convert mass and molar units from one another. Okay. Next, the result should clearly state the therapeutic concentration of the drug tested. It must be remembered that different indications for therapy, age, or ethnic differences, difference in pharmacokinetic or pharmacodynamic could result in different therapeutic ranges being appropriate for different population groups. Okay. Hence, critical assessment of the original literature and census recommendations for therapeutic ranges should be encouraged. Okay. Last is your clinical interpretation. Okay. So, the information which is basically required to interpret the drug concentration, it includes time at which blood sample was taken, time at which dose was given, dosage regimen, patient's demography, co-medication, indication for monitoring, pharmacokinetic and therapeutic range of the drug. Okay. So you need to compare the measured drug concentration to the established therapeutic range. Consider the patient's clinical response, comorbidities, and other factors that may affect the drug disposition. Interpret the result in conjunction with the clinical judgment and available evidence. Okay. Lastly, we have got conclusion. Like what you will conclude once all the things are been done. Yes, you need to give a regimen to the patient, right? So once the report is in your hand, based on TDM results and clinical assessment, you need to make dose adjustment as necessary to achieve optimal drug concentration within the therapeutic range. Consider patient specific factors including age, weight, renal or hepatic functions and concomitant medication. The story doesn't end over here. Regular monitor and patient clinical response, drug concentration, and potential adverse effects. Okay, you need to monitor these things regularly. You need to adjust the dose or dosing interval as needed based on the present TDM and clinical evaluation. Okay, so these all are the protocol of your therapeutic drug monitoring. I hope you people found this video helpful. Thank you for watching.